Good morning, everyone. Before I get started, are there any questions about the course or the content from the previous classes? Okay, everything's clear. Good. Um, Let me share my screen and get started then. So a couple of reminders. Quiz 1 is available and is due on January 29th, which is a Friday. Quiz 2 opens immediately after. It opens at midnight on January 30th, and it will be due on Friday, February 5th. Okay, that quiz will test you on the chemistry of life, and make sure to rewatch the videos and the supplementary videos that I included in the quiz description. And there is a new participation assignment that is due today. So today's lecture is going to be on the energy of life. And there's going to be 2 questions in the quiz created as a participation assignment. Please take it before midnight tonight. Should not take long for you to complete them, okay? As I mentioned, today's lecture is going to be on the energy of life. First, what is energy? The operational definition of energy is the ability to do work. So simple enough. If a cell or an organism is not able to do work, then it has no energy. Energy is required for carrying out chemical reactions. So a lot of chemical reactions will simply not go if no energy is input into the systems. Energy is required for transporting molecules across the membranes. We'll discuss today the different ways how molecules are transported through the membrane. So last time I discussed the structure of the cell, the components of the cell, and one of the cell components, the important one, is the cell membrane, which protects the cells from external factors as well as molecules that maybe should not enter into the cells. So some molecules, remember, can pass through the membrane freely. That they are small non-polar molecules, such as CO2, carbon dioxide, oxygen. Water is polar, however, it's small and it can pass through the membrane. What else? But ions and large charge molecules, they are stopped by this head portion of the phospholipid membrane. So these guys need to be somehow transported into the cell because cells need potassium, cells need potassium ions, cells need sodium ions to function. So the energy is required to overcome this barrier of the cell membrane and import these ions into the cell. What else? And maintaining internal constancy or homeostasis. What I mean by that is maintaining constant temperature requires energy. Maintaining constant concentration of any solid or any substance requires energy. Particularly if this concentration is out of balance with the outside surrounding concentration of the cell. So without energy input, things start to fall apart. For instance, plants use sunlight for energy. Okay, there's chat somewhere. Ansley, are you here?
Yep, I'm here.
Great. Could you please monitor the chat?
Mm-hmm.
And interrupt me if the question cannot, if you cannot answer the question. Okay.
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 Is it multiple choice?
The
Participation assignment for today.
Yeah, one of the questions is drop down. It's similar to multiple choice, and the other one, I think it's multiple choice, but both of them require interpretation of images or graphs that will show today. All right, back to the energy of life. So, as I said, without the energy input, things start to fall apart. If the plants use sun for energy, the energy of sunlight, And if you place a potted plant into the dark room, you will see that it will wilt fast and it will die. So it is not able to keep alive and maintain the reaction it needs for life without the energy input or the input from the sun. Similarly, what happens when we stop eating? We gradually also fall apart. First, our body digests itself, and then we die because the without energy input, we cannot survive. The chemical reactions inside our bodies cannot be maintained. So in this picture, it's pretty simple. It's for you to review later that the biker may have eaten ice cream before going on the bike ride. And if you eat lots of carby foods or sugary foods, you will have more short-term energy for intense workouts, such as a bike ride. Energy has different forms. The two forms of energy are kinetic energy and potential energy. Kinetic means motion. So kinetic is a Greek word. Kinesis means motion from Greek, I believe. There's, as a side note, there's a lot of Greek words and Latin words in science. So if you have knowledge of Greek or, well, Latin, if you went to that kind of school, you will have less trouble with remembering these terms. So kinetic energy is the energy of motion or movement, and potential energy is the stored energy that is available to do work. For instance, consider a biker going uphill and then downhill. On the uphill, there is a lot of potential energy that the biker has. And when the biker is going downhill, there is a lot of kinetic energy from the movement downhill. Similarly, if you have a ball and a spring, if you compress the ball and just before it's released from the spring, it has a lot of potential energy or the ability to do work, okay? And once the spring is released, once you stop the ball from being pushed upwards, the extended spring releases the kinetic energy, and this energy is transferred to the ball. So the spring is released and pushes the ball upwards, and this push of the ball is considered kinetic energy. All right. In biology, those are kind of physicky concepts there. But in biology, we consider potential energy, or not consider, potential energy is stored in chemical bonds. So here you see the sketch of the glucose molecule, and glucose is the table sugar that we eat is contains the glucose. I believe it's just glucose. So it tastes sweet, okay? It's just think table sugar if when you see the glucose molecule. So it has high potential energy because it has all these covalent bonds that can be broken. Now, this potential energy stored in these covalent bonds, when the bonds are broken, the energy is released and can be used and is used by the live organisms to carry out work. And this work can be riding a bike or maintaining chemical reactions, importing substances into the cell and out of the cell. So all the various ways of work, all the various types of work are performed because these covalent bonds are broken down. So yeah, this is basically the summary of what I just said, and for you to go over later. Oh, right. So if the cell cannot capture the energy as it is released, the energy will be lost as heat. So no energy transfer is 100% efficient. The energy, part of energy that is released from the breaking down of chemical bonds is always released as heat. And this is kind of a bad news because heat is still energy, but it's more disordered. It's a disordered kind of energy that cannot be harvested. So if you lose the energy as heat, you cannot reharvest it and use it to carry out any additional work in the body or in the live organism. So there are two laws of thermodynamics. If you took physics in school or physics in college, you may have encountered them. We're not going to go into second law of thermodynamics, but the first law is kind of important to keep in mind. And the first law of thermodynamics states that energy is never created or destroyed. It's being transformed from one form to another. So energy changes from within the biological system. This bit here shows the energy currency of the live organisms called the ATP molecule, and I will explain more about it later, and we will discuss some ATP generating and degrading reactions later in very high level terms, okay? But important thing to remember as we live on planet Earth is that all life on Earth depends on sunlight. Sun's energy is kinetic. So here's our sunlight here. Sunlight energy is kinetic. And does anybody know why the sunlight energy is kinetic? Just feel free to unmute yourself if you know why the sun's energy is kinetic.
Is it because photons?
That's exactly right. So the sunlight is actually the movement of these tiny particles called photons that are smaller than a nucleus, okay? So these tiny particles called photons, they move in a wave-like motion, and this wave-like motion creates kinetic energy, or is able to provide kinetic energy to the living organisms, namely plants, because they are the only plants and algae are the only organisms that can harvest the energy from sunlight and transform it into sugar. So the plants capture the sunlight energy via the process called photosynthesis. Photosynthesis is the conversion of the sunlight energy, water, and carbon dioxide into oxygen and sugar, or glucose, which is then transformed into cellulose to construct cell wall and various other biological components. So sugar is consumed by heterotrophs, and you should know what heterotrophs are from the biodiversity lecture. So heterotrophs, they consume sugar, and they use some energy stored in these covalent chemical bonds, and a lot of energy is lost as heat. So once the heterotrophs consume sugar, part of energy is lost, and they convert sugar into carbon dioxide and water. So this whole energy cycle, this whole energy creates energy series of consumptions. Pardon my formulation, creates a nice little loop here. So yeah, something I needed to mention here. Something interesting about heterotrophs. And it's gone. Okay, maybe it'll come later. Oh, right. So again, to reiterate, the energy transfer is inefficient, and I remembered a bit of information that I wanted to share. So way back when I took ecology, we were taught that the rule of thumb is about the efficiency of the energy transfer was 10%. So heterotrophs are able only to use 10% of the energy stored in the primary organic matter, or the organic matter in plants. Okay, if let's say we have a deer who eats plants, and only 10% of energy can be utilized by deer that is stored in plants. Let's say a wolf eats a deer, and a wolf is able to only to capture 10%, use 10% of energy that is stored in the organic matter of a deer, in a deer meat, okay? So that was a bit of information that I wanted to share. But the plants are a bit less efficient than that. Plants are able to capture, I believe it's like 1 to 6% of the energy that is incoming with the sunlight. All right, so chemical reactions can require or release energy, and it is kind of intuitive when is energy required in a chemical reaction, and when is it released. For instance, think of look at this picture here, like building a house. You build something complex out of simple components. So when you build something complex out of simple components, you need to input energy. So lots of labor is required to build a house. So similarly, like in biological chemical reaction, if you're synthesizing or building a large complex molecule out of simple molecules, then you need an input of energy from somewhere, not heat energy, remember, because heat energy is disordered, it cannot be harvested. To build this more complex molecule. An example of the biological reaction that requires energy is photosynthesis. In photosynthesis, 6 molecules of carbon dioxide are combined with 6 molecules of water. In the presence of the sunlight energy, they are converted into glucose and 1 molecule of glucose and 6 molecules of oxygen, which, by the way, we breathe. Now, if you have a complex molecule and you want to break it down into simpler components, it is typically a reaction that gives out energy. So if we continue our analogy with the house, if you demolish a house, there's a lot of kinetic energy created. Okay, so and this energy can be harvested for other reactions. So in our case, when we eat sugar, it is a complex molecule. We ingest a lot of potential energy, and our bodies are able to break down that complex molecule of glucose and use the energy of breaking of these covalent bonds to perform work. Okay, so the example of the biological reaction is what I just described. So this reaction is essentially the reaction of oxygen with glucose. The bonds in the glucose are broken down, and we get 6 molecules of carbon dioxide and 6 molecules of water. So this is the process. This process is called respiration, aerobic respiration. And this is basically a summary of how we break down water expressed in chemical terms. So some chemical reactions, they transfer electrons. Remember, electron is the tiny bit of it's a component of the atom. Atom consists of nucleus and electrons. There can be one, there can be more than one, depending on which atom we're looking at. Excuse me. So when the atoms transfer electrons to one another, this type of reaction is called oxidation reduction reaction, or in short, redox reaction. And most energy transformation in organisms occur in these redox reactions. So the process of oxidation is when an electron donor molecule loses its electrons. Atom, not molecule, atom. When an atom loses its electrons, it is considered oxidized. When an atom gains the electrons, it is considered reduced. Well, it says molecule here, I guess it's true for molecules too. Yeah, so the process of losing of electrons by a substance, by a molecule or an atom, is called oxidation. And the process of gaining electrons is called reduction. Molecule that has lost its electrons is called oxidized. The molecule that has gained electrons is called reduced. Okay, let me check my notes to see if I'm missing something important. So the reduced molecule gains the energy stored in the electrons, and the oxidized molecule loses the energy stored in the electrons. So the electrons are the bit that transfers energy, kinetic energy, okay? So in the yeah, go ahead.
Sorry, Dr. H, can you give an example of a transfer, like an electron transfer? I can't think of one.
Sure. So in here, in these biological reaction, here, atom of, let's see, O2 here is oxidized. It loses the electrons. And here, oxygen is reduced. It gains the electrons. So this is a quick example, on the spot example that I can come up with. So I'm sure we'll come. It will come across. We'll come across the more colorful examples later on. But for now, it's important to remember that a molecule or atom that gains electrons is being reduced, and the molecule that loses electrons is being oxidized. And the electrons are bits that contain this kinetic energy that is ability to do work. Okay, in the early days of chemistry, oxidation was defined as a gain of oxygen atoms. For instance, when iron is oxidized, it turns into rust. It changes color. It obtains. It starts showing this red residue on the top, and it's considered to be corroding. So, but later on, it was discovered that the change in properties of substance with oxygen addition actually involved electron transfer. So in the reaction, here's an example. In the reaction, when iron reacts with oxygen, so iron Fe plus O2 from the air, iron gives up electrons and oxygen gains them. So the reaction is elemental iron plus O2, and it will turn into a ferrum oxide or iron oxide. So iron loses the electrons and oxygen gains them, and this is considered a redox reaction. So, and again, this is a bit of a chemistry history here, because oxidation kind of implies that the oxygen is involved, but the catch is not always. Oxygen does not have to be involved in the reaction for it to be considered a redox reaction, an oxidation reaction. It's just an atom or a molecule has to lose an electron or more than one electron and to be oxidized. Any other questions, Ensley?
No, I think we're good.
Okay, good, thanks. So as I mentioned before, oxidations and reductions occur simultaneously, and because they occur simultaneously, the reactions are called redox. So in this picture here, you see the electron transport chain, or a series of proteins embedded within a cell membrane. And these proteins are participating in sequential linked oxidation reduction reactions. And they are one of the mechanisms that the cells gain energy. So the reaction is started with a molecule that acts as electron donor, or the molecule being oxidized. So it transfers electrons to the first protein. The purple bits embedded in the membrane, again, are proteins that are a part of electron transport chain. So the electron gets on the protein, and the protein is being, this bit is being reduced. and then it goes to this protein, and as this protein as electron moves into this protein, now this protein is being reduced, and as this protein is being reduced, this protein is being oxidized, and this electron moves from protein to protein, and when an electron lands on the protein, it is being reduced. When the electron leaves the protein, it is being oxidized. And every time the electron jumps from protein to protein, some energy is released. And finally, once the electron is at the end of the electron transfer chain, it is transferred to the electron acceptor molecule, or the molecule being reduced, like the end acceptor molecule. So, and you will see that the potential energy of electrons, it goes from high to low, because as the electron jumps from protein to protein, part of the energy is lost or is released into the cell for performing some work. So ATP is an important energy currency molecule in the living organisms. What ATP stands for is adenosine triphosphate, and chemically it is a nucleotide that temporally stores energy. Does anybody remember what other nucleotides are there? Like what other important molecule consists of nucleotides, or molecules, plural? DNA and RNA. DNA and RNA consists of nucleotides. So ATP is really the form of energy that the cells use. So when glucose is broken down, these energy from the covalent bonds are used to generate ATP, adenosine triphosphate. It's getting more complicated as we get deeper into it, but that's just how it is. Okay, so the simple portrayal of things is you eat sugar and you gain energy from breaking down of the covalent bonds. But the more accurate view of things is you eat sugar, these energy in the stored in the covalent bonds of the sugar molecule is used to create ATP, and then ATP is used to perform work within the cells. All right, so the bond with the high potential energy is right here. So ATP has 3 phosphate groups as implied by its name. And one of the phosphate groups is, when one of the phosphate groups is lost, a lot of energy is released that can be used to perform work. Okay, when one of the phosphate groups is lost, intuitively, you can kind of guess what kind of molecule is created. When one of the phosphate groups is lost, adenosine triphosphate turns into adenosine diphosphate. So ATP turns into ADP when this one phosphate group is lost and energy is collected from the ATP, from this high energy bond. So ATP releases stored energy, the energy stored in that high energy phosphate bond. What is it? Oxygen to phosphorus bond. And removing the endmost phosphate group by hydrolysis releases the potential energy stored in the ATP. And the cell uses this energy to do work. So this is a schematic hydrolysis reaction, which you may or may have know about already, because there is a supplementary video about hydrolysis reaction, and I believe I will test you on it on the second quiz. So what happens is the water it participates in breaking down of this phosphorus to oxygen bond, and one of the phosphate groups is lost, and what remains is ADP, or adenosine diphosphate, and energy that can be used to perform work. And the process of breaking down of chemical bonds with the help of water is called hydrolysis. So hydro means water, and lysis means like breaking down. So ATP is formed during cellular respiration. And cellular respiration is a series of chemical reactions that release energy from sugar, producing ATP from ADP. So let's say we eat an energy bar, and we gain a lot of potential energy because energy bars typically contain a lot of sugar. And from this sugar, ATP is formed, and ATP is broken down by hydrolysis. And once it's broken down, it releases energy, which we then use to ride a bike or exercise more vigorously or just breathe, because that also requires energy. And so this ATP is turned into ADP and phosphate group, extra phosphate group that just kind of hangs around because energy is never created or destroyed. It's transformed from one form to another. So this phosphate group is kind of around after it was lost from the ATP. And then this energy from more sugar is used to add this extra phosphate group to ADP, turning it into ATP again, and the cycle repeats as long as you supply your body with energy. Okay, so what else I wanted to mention here is that chemical reactions in cells must occur very quickly to sustain life. So it's like sub-second kind of speed. And often, so an important reaction is this hydrolysis of ATP. And often it's, a lot of reactions are kind of inefficient. They don't occur at the speeds needed. For instance, if we move away from this graph for a second, imagine a piece of paper, which is essentially cellulose. And cellulose is a polymer, and its monomer is glucose molecules. So a molecule of cellulose it consists of multiple glucose molecules. And imagine we put a piece of paper into the water, and we try to perform hydrolysis on cellulose, which should break down this polymer into sugar. And it's not going to take a short time to do that. I mean, it can take a while for a piece of paper to degrade. And this is not okay for on the biological level, because we need this energy quickly. We need to break down the polymers into monomers and harvest the energy quickly. So what helps to speed up the reactions in the biological systems is enzymes. So an enzyme is a protein that acts as a catalyst. And what a catalyst means is a catalyst means, sorry, I'm blanking on it here, but something that speeds up the reaction. It's literally something that speeds up the reaction, and it's kind of a synonym to enzyme. So
Catalyst?
Catalyst, C-A.
You're welcome.
An enzyme, it speeds up a chemical reaction without being consumed. The enzymes kind of hang around there. They help the reactions occur, but they themselves are not digested typically. Eventually they are, but as long as they're useful and not damaged, they help the reactions to be quicker. So enzymes convert reactants into products. So in a chemical reaction, we have a substrate, or a substance that an enzyme acts on. We have reactants, or molecules present at the start of the chemical reaction. If you're thinking about a chemical reaction in terms of a chemical equation, so the reactants are located in the left part of the equation, and the products are located in the right part of the chemical equation, and they're defined as the molecules present at the end of the chemical reaction. And this is kind of a simple depiction of how enzyme works. So enzyme is a protein, and proteins will be consistently depicted in purple. So we have an enzyme protein, purple protein, an enzyme which speeds up reaction, and we have a substrate or a molecule to be broken down. Let's say this is cellulose, the polymer of glucose. So cellulose binds to enzyme at the active site. So this active site, the substrate and the active site fit together like a jigsaw puzzle, okay? And once the substrate is bound to the active site, it forms the enzyme substrate complex, and eventually water comes in. And the enzymes are broken, the substrate is then broken down into products and kind of fall out of the active site. So if this is cellulose, these two guys are two molecules of glucose. An important part or an important property of a chemical reaction is called an activation energy. So the activation energy, as long as we're talking about energy, is the energy required to start a reaction. So what some reactions take a long time to get started, and some reactions are pretty fast. But usually biological reactions without enzymes take pretty long time to be carried out. So this graph shows the progress of reaction with and without enzyme. So so the x-axis here is the progress of reaction, the y-axis is the potential energy of the molecule, and we see that with enzymes the potential energy is lowered. So activation energy is lower. So when activation energy is lower, it is a good thing. Okay, it means that reaction will go faster. But when potential energy is high, there can be it can be either a long time for the reaction to be performed, or it's not going to go because there's no energy to start the reaction. What else? I wanted to say this. Oh, about enzymes. So an example that I like about enzymes working on helping speed up the reactions is we have an enzyme in our saliva that helps breaking down complex sugars into simpler sugars. So for instance, let's say you take a piece of rye bread, which contains a lot of complex sugars, and you kind of chew it for a while. So if you don't chew it for like 20 seconds, if you chew it for like 2 minutes or so, the rye bread will start tasting sweet. And it is because the enzymes helped start the reaction or lower the activation energy of the reaction in our mouth, and help the enzyme in our saliva broke down the more complex sugar in the rye bread into a simpler sugar. And as a result, the rye bread starts tasting sweet after 2 minutes. The enzyme activity is affected by physical and chemical factors. Okay, so physical factor that affects enzyme activity is temperature. So it's one of the common things that affect enzyme activity. So most enzymes are active within a range of within a certain range of temperatures. So enzymes are not active if it's too cold, and enzymes are not active if it's too hot. And of course, there are various types of enzymes. So the enzymes in a human body will have very different range of the active temperatures than enzymes in, let's say, a bacterium that lives in a hot spring, which is the comparison given here. So enzymes have the optimal temperature at which their activity is the greatest. However, if the temperature gets too high, then enzyme being a protein, the protein starts breaking down after a certain temperature is reached. And this process of breaking down of the enzyme when the temperature is too high is called denaturation. So like denaturation is the process of breakdown of enzyme when the temperature is too high. By the way, this is the reason why if we spike a fever over 42 degrees Celsius, we simply start dying because all the proteins in our body are start being denatured. So if you have, that's why if you have a high fever, like 40 degrees Celsius, it is doctors usually recommend that you break the fever, that you lower it with medication to prevent the fever spiking too high, and to prevent the denaturation or breaking down of the proteins in your body. All right, so back to the graph. The enzymes from humans, or enzymes that we contain in our body, they have an active range from about, 25 degrees Celsius to about 42 degrees Celsius. The common human body temperature is about 37 degrees Celsius, and 36.6 is considered a healthy one. And so on x-axis we have temperature, on y-axis we have the rate of reaction, and we see that the rate of reaction increases from the 25 degrees Celsius to 37 degrees Celsius. It kind of starts falling after 37 degrees Celsius, and it rapidly drops off at about 42 degrees Celsius. And it is because the enzymes start breaking down because the temperature is too high. High temperature starts modifying the structure of the protein. Like if it helps you, think of an egg that you cook. So an egg has protein, a lot of protein. So when you crack it and throw it on a skillet, it becomes cooked, or the proteins become denatured, and you can actually see the clear white protein turn it into white stuff. Now for this bacterium that inhabits the hot springs, just think of an abstract bacterium in a hot spring. The enzymes have a very different range of temperatures under which they're active. So the enzymes start being active at about 55 degrees Celsius, which is pretty hot. And then the rate of reaction increases, meaning the enzymes are more effective as the temperature increases. The temperature peaks over here, and then after a certain temperature, the enzymes start getting denatured, break down, and no reaction goes. The organism dies when no reactions start happening. When the reactions stop, the organisms die. All right. Any questions about that? Ansley, were there questions that needed my assistance?
Nope, just going over the graph again, which you did. So
Okay. Cool. Okay, we're good on time. So in the very beginning of the lecture, I mentioned that energy is required to perform work. And work can be like, we may associate work with physical exercise, but work occurs also on a cellular level or subcellular level. And I want to talk about that for a bit, because a substantial amount of energy goes into import and export of various substances from and to the cell. So a cell's interior, or inside of the cell, is chemically different from its exterior, or the surrounding environment. So the cell maintains this difference by regulating transport of dissolved substances, which are called solutes, across its membrane. So this is kind of important. The I will refer to dissolved substances in the later slides as solutes. But not all of the methods of transport of solutes across the cell membrane need energy. So next I will go over various ways the cells use to import and export the solutes into and out of the cell, and classify them as ones that require energy and ones that do not require energy. So first, let's figure out. The first method that cells import and export, by which cells import and export molecules to and from the cell, is called transport by diffusion. Okay, so diffusion will happen if there is a small nonpolar molecule, and if there is a gradient in concentration of whatever solute we're working with, or whatever substance we want to import or export out of the cell. So let's figure out what this concentration gradient is. Picture imagine you want to make some tea, and you pour hot water into the cup, and you drop a tea bag in, and you can observe that the solute, or the molecules of tea, which are brown colored, right? They at first they appear close to the tea bag, but as the time passes, they occupy the whole volume of the cup. So in this case, in the very first case, because there is more concentration of the tea molecules around the tea bag than closer, like to the surface of the cup, let's say, there is a gradient in concentration of the tea molecules. But when we have the situation of the tea molecules spread out evenly across the cup, across the water in the cup, there is no gradient anymore. And the reason why the tea molecules spread around the cup is through process of diffusion, and tea molecules kind of go down a gradient of concentration, meaning they go from the area of high concentration of tea molecules to the area of low concentration of the molecules. The process of movement of tea molecules, or any molecules in between other molecules, is called diffusion. And I believe Dr. Hoffman covered that atoms move around and molecules move around kind of chaotically. And eventually, because of this chaotic movement, the tea molecules that were concentrated around the tea bag will be spread out evenly across the cup, because molecules move chaotically against each other, and they kind of move in between each other's spaces and kind of delete this gradient of concentration. So simple diffusion does not require energy. And the simple diffusion occurs when the solutes go from the areas of high concentration into the areas of low concentration. So when a solute or a substance goes from area of high concentration to an area of low concentration, we say that molecules move down their concentration gradients. To move down the concentration gradient is to move from the high concentration to low concentration, from the area of high concentration to an area of low concentration. Simple diffusion is a type of passive transport, and it takes place when the concentration difference on one side of a membrane is compared to the other. So this is basically a summary of what I was just talking about. Now, water. Ooh, that's kind of tight. Water has its special way how it moves around. It moves around by osmosis. Osmosis is the process that does not require energy, the process of penetration of water through a semi-permeable membrane that does not require energy. So it doesn't take energy for a cell to absorb water or to release water. And it's considered also a type of passive transport. So it takes place when there is a different concentration of water on one side of the selectively permeable membrane compared to the other side. So let's say the water molecules in this picture are red, and the solute molecules are, well, blue and green. So if the water, here the water is, there's more water than the solute, so the water will move down the gradient of concentration. And when water moves down the gradient of concentration is called osmosis. Okay, I will stop here because I still have a few slides to go over. I will go over them on Wednesday, and which is good because photosynthesis lecture was kind of short, right? So I'll stop here, And we have a remaining minute for questions.
So it looks like someone's asking if you were still uploading the PowerPoint even though we didn't finish the lecture.
I will. Yeah, I will upload what we went over today, and I will upload the slides that we did not go over today on Wednesday, along with the slides from photosynthesis. Okay, so would you mind go over simple diffusion and osmosis? All right, so simple diffusion, let me reshare here quickly. Simple diffusion is when, let's say, a simple molecule, a simple nonpolar molecule, let's say oxygen is a simple nonpolar molecule. Oxygen moves in between the molecules of the membrane into the area of the cell. This is a specific biological example, but in case when we don't consider a living organism, the process of diffusion is simply the movement of molecules in between spaces of other molecules. So here, the process of diffusion is basically movement of T molecules in between the spaces of the water molecules. And for osmosis, it's diffusion, but for water, okay? So it's when water molecule moves down the gradient of concentration through a semi-permeable membrane. So if water concentration is high on one side of the membrane, then water will move into the other side of the membrane simply because there's less water on the other side of the membrane. So osmosis is the movement of water. Diffusion is a more general term.
For the participation quiz today, we won't have any of the questions that we didn't get to, correct?
No. All questions we covered for the participation quiz. Okay. Any advice on which medium to focus on the most when preparing for quizzes? Obviously, everything I assume, but which medium tends to cover the content? Okay, for quizzes, I post what you need to go over, and everything that I post will be covered on the quizzes. So I thought it was pretty okay and helpful for preparation. All right, guys, we're over time, and I'll see you on Wednesday. Thanks for joining in, and have a good week.